Hello, I'm Tim McClanahan. I'll be presenting the findings from a one-year project to understand the distribution of biodiversity in the Western Indian Ocean. This study is funded by the USAID and the Department of Interior as part of a project to prioritize funding for conservation and management in the Western Indian Ocean. The Western Indian Ocean is a tropical location with high biodiversity, many coral reefs. It's peripheral to the center of biodiversity in the coral triangle of the Western Pacific, but it also has very high biodiversity, but has not been studied as extensively as many of the areas in the coral triangle. Most of the studies as well have focused on two ways to identify the abundance of species or species richness, that is overlapping species distribution polygons, that is to use sightings of species in different locations and map out their distributions by connecting the points of their identification. The second method is cumulative level observations, what is called rarefaction, which is the cumulative numbers of species as you sample across different areas. Both of these have been used in our region to identify areas of species richness. They suffer from a number of problems that I'll discuss. One of the main problems is the intensity of sampling. If sampling is infrequent in many areas, then it's very difficult to develop both polygons or rarefaction techniques, and that's true for much of the Western Indian Ocean. There's been much more intensive sampling in the Coral Triangle or in the West Pacific and also within the Caribbean, and therefore those areas probably have better estimates of biodiversity. Consequently, there's a need to consider new ways of estimating richness of species in the Western Indian Ocean, and one of the primary aims of this project was to develop modern techniques to estimate biodiversity when there is not a large amount of sampling done by professionals such as museum collectors. The paper here evaluated a large amount of this distribution data, and they came to the conclusion that the Western Indian Ocean had been undersampled and maybe one of the high biodiversity centers in the world, but it's difficult to know due to the undersampling problem. The area that I'll be discussing includes 9 ecoregions and 11 nations. They're shown here. These ecoregions have distinct faunas. They're closely related, but there are certain environmental conditions and slight differences in the fauna, and that makes them unique for their distribution. So much of the analysis that I'll do is both done at this ecoregional level and at the national level in order to determine where this distribution occurs and to what extent these national or ecoregional delineations influence the species richness. We have mapped out the reef cells in each of these ecoregions and countries, and you can see sort of the number of these 6.25 square kilometer cells in each of those ecoregions. The big ecoregions are the East African coast from southern Kenya to northern Mozambique, and the second is the western and northern Madagascar, which includes the western part of Madagascar, include the islands offshore from Mayotte to Comoros. A number of the other ecoregions are considerably smaller in terms of the number of reef cells, and this distribution of reefs has an effect on the biodiversity, as we'll discuss later. One of the goals of this project was to reconcile some of the past prioritization exercises that have occurred in this region. We had the East African Marine Ecoregion program in around 2004, and then more recently the Southwest Indian Ocean Fisheries Project in 2015, which identified a number of potential priority sites for conservation. One of the problems that you could observe just from these publications is that they often didn't overlap particularly strongly, and that the decisions were often based on sometimes anecdotal information, sometimes not the best science or quantitative information. So one of the goals of this project was to develop more quantitative approaches to biodiversity predictions using modern methods, statistical methods and other methods, and to see to what degree these potentially high biodiversity areas might have in terms of corresponding with previous prioritization exercises. The project has three main components of it, which is the biodiversity aspect that I'll be discussing today. There's another part of the work that's an institutional analysis, which looks at the institutions in the regions, the threats and their preparation for these kind of disturbances, climate and otherwise. And then there's a review of the policies, laws, and regulations and cultural norms in each of these countries. Once we combine that information, the idea is that they will identify priority focal areas, and from the situation analysis we'll be able to determine what the main threats are, and that will guide priority actions. The project team, the core team, involves the following four people. Myself is focusing mostly on the marine biodiversity assessment and threats. My main collaborator is Maxwell Azali, who has a background in spatial modeling and statistics. Yawida Mathiga is leading the institutions and protected area and fisheries and coastal and development issues in the Western Indian Ocean, and she's assisted by Carol Abunge. The table here summarizes sort of the past scientific publications that have evaluated biodiversity in this region. And one of the main points is that it's generally focused on corals and fish, marine turtles, sharks to some extent, or it's a part of a global analysis where species presence-absent data has been used from this region to do global mapping of biodiversity distributions. While these have been very useful, they suffer a number of problems. One is that they generally produce very coarse scale resolution patterns in space. In many cases, they're looking at many sites over very large areas, and many of the recommendations that come out of these studies are to identify very large areas of coastline. It might be the entire northern Madagascar, the coastline of Tanzania. So it's very difficult to know where within these larger areas you could pinpoint this information. So one of my goals was to reduce the resolution, to create finer scale evaluations of biodiversity that might be more helpful to the current trend towards localized conservation, often done at the community or often even county levels. Another issue around past studies is that they have typically focused on charismatic species. Much of the work has been based on what you can observe from land or boats. That often results in focuses on reproduction, nesting, and calving. Oftentimes it also focuses on large body sizes and also species with large ranges and with a lot of overlap with other species. So these create somewhat of a bias in terms of selection of sites, and yet the total biodiversity may not be necessarily correspond well with these particular aspects of species assemblage and distributions. So one of the goals as well was to ask whether or not the prioritization processes has been biased, and to what extent it may not have included the most species-rich areas within its investigations. So in order to improve on the situation of limited number of taxa, we still had to use some of the taxa for which there is sufficient data, and essentially that boils down to coral and fish. So I contacted the various people, listed here on in this table that have collected coral and fish data. Many of them have used methods not dissimilar from the methods that I and Nyawita use, and many of them were very keen to share their data and participate in this project. So the result was a considerably large data set and very widespread distribution of coral and fish data. The idea here, though, was not just to predict coral and fish diversity, but to use those two groups as a proxy for total diversity. There's a number of scientific advances that make this work possible. That is to map biodiversity on very large scales. One is the development of moderate resolution maps of habitats such as coral reefs. In our region, there are 7,039 cells or mapped coral reefs on the scale of 6.25 square kilometers. We know their locations, and we can use those locations as a basis for estimating biodiversity. There's more people in the field collecting data, oftentimes using similar methods, and we're more able to communicate with each other due to improvements in media, cloud data sharing, and all these other technologies that make it easier to work together. There's also complete satellite coverage of, and from that satellite measurements, we have a number of environmental variables and proxies that are being produced from the satellite data that are very useful for estimating the state of the ocean. And fourthly, there's an increase in the capacity for doing analysis with machine learning or artificial intelligence. These allow us to do much more sophisticated analytical evaluations of data that surpass the capacity of the human mind. So the specifics of this modeling involved the data we collected on species richness in corals and fish. We used a search sampling method and obtained 1,000 observations from ourselves and our collaborators. And we obtained 1,200 observations on a number of fish in 6 families. These families are known to be highly diverse species and good predictors of total diversity of coral reef fishes. We compiled 70 environmental data bases or layers. Each of those are spatially complete. That means that all 7,039 cells have the basic information on that particular environmental, human, or geographic variables. For instance, there could be temperature, mean temperature variable, variations on temperature. There are many layers, and that allows us to make predictive models based on many environmental sources of information. The analysis based on machine learning, statistical analysis, we use the Boosted Regression Tree method. This deals with complex relationships between environment and the data you're modeling. And it allows for seeing the distributions of these relationships, which you'll see later on are often quite complex. We can also develop what we call ensemble or multiple models. That is, rather than relying on a single model, we can look at multiple models and ask whether the type of model we choose has a big effect on the outcomes, or whether, and also to combine models in order to get sort of an averaged output for estimating, in this case, species richness for all of these 7,039 quadrats. The study sites are shown here on the map. It's a widespread distribution. Much of that was collected over the last 20 years, various sampling trips, and also collaborating with people who work in these different environments. So the data is quite well distributed in the Indian Ocean, which would reduce some potential biases of limited spatial sampling. Here I just wanted to show you some example of these environmental layers. This is the distribution of a variety of environmental variables. On top of some of these common ones, such as temperature, chlorophyll concentrations, etc., we also identified each cell for the type of fisheries management in each of the cells, and we used 4 categories of unrestricted fishing, restricted fishing, high compliance closures, which are only 2.1% of all cells, and low compliance closures, which we are 14% of the cells. So these 6 layers shown here are just representative of 70 layers that we looked at for the total modeling exercise. So one of the things you can do is identify the importance of these variables, and you can create more than one model. So if you find 2 variables that are closely related to each other and you would like to eliminate one because they're redundant with the other one, you can put it in a second model and use it in that model. And by that way, we create these ensemble models where you can have final models, which are the average predictive performance of the average of the 2 models. So here's an example of fish diversity, species richness, 2 different models that remove some of the highly correlated variables. But what you can see is, in general, these models do about the same, but there's some that have stronger effects of some variables. In both cases, for example, you can see that the total biomass of fish is one of the strongest predictors of the number of species of fish. And then there are a number of other lesser important variables that will have smaller influences on the model predictions. One thing that we wanted to do was not just to focus on fish and corals, but to actually get an estimate of total diversity. And so these coral and fish diversity is essentially a proxy for total diversity. But in this case, you can see we've correlated the numbers of coral taxa and number of fish taxa, and you can see that they are positively correlated. There is variation around that line, but the good thing for our purposes is that because they are positively correlated, the sum of those 2 taxa is probably a good indicator of many different taxa. Specifically, you can be confident because corals have different life histories than fish. They live in different ways, so the environment is expected to have big differences in terms of the environments in which they do well. But they're not so different that they're not positively correlated. So this is a good finding that gives us some hope that our combination is some measure of reflecting many of the other different types of species, whether it's seagrasses or mangroves or snails or copepods, etc., etc. So this here would show you a partial effects for the fish number of species. You can see there's a number of variables, probably 9, that are most influential, but there's as many as, in this case, I think 26 or 28 variables that have some influence on species richness. And in the bottom right graph, you can see sort of the relationships between these variables and numbers of species, and you can see that they are sometimes complex, but they show some predictable patterns. For instance, the number of species of fish rises very quickly with fish biomass and then levels. The other relationships are somewhat more complex. Some are what we call unimodal or hump-shaped, but each of these relationships is accounted for in the final model. Here's the same responses for coral taxa. It's a different group of variables that are important here. Some of them are related to temperature, including degree heating weeks or cumulative excess heat, the skewness of the temperature, the kurtosis in terms of the shape of the temperature distributions, wave energy, dissolved oxygen, salinity, etc. So this gives you an idea of what's influencing corals, and it's really not the same as fish. There is some overlap, but in general, they are affected by different environmental parameters. So you may think of them as potentially not occupying the same space, but in fact the positive correlation suggests that they are related in space. So this is the fit of the data to the empirical data. The issue now is that once you have this fit, if you like, to the the empirical data is to use the same information that you use to fit this curve in these cells where you have no empirical data. So you may have around 600 cells with data, but you need to fill out and map the additional 7,039. So because you have this environmental data for those other cells, you can use those numbers to calculate what actually would be the predicted fish, coral, and the sum of the two richness measurements in these other areas. So here we show some of the maps of the distribution of biodiversity. Corals on the left, fish diversity in the middle, and the combined the both species on the far right. And then we break them into different categories, 6 different categories, as to whether they fall into the lowest or the highest, low being red and high being green. One thing that we've done here to remember is that we've normalized some of the things such as fish biomass. We know fish biomass is very important for biodiversity, so to compare sites you have to solve it for that one particular biomass. In this case we picked 600 kilograms per hectare, which is above the saturation curve. So this allows you to then look at biodiversity in a comparable way without predicting what might actually occur there. So in many cases what you're seeing is the likelihood of biodiversity in the absence of certain human impacts such as heavy fishing. This allows for a fair comparison of sites across all of these 7,039 cells. Another issue for making fair comparisons is the spatial delineation of the distribution of the data. That is, at the provincial level, you have to compare all sites to all sites in the province, and that can create useful information for knowing what is most diverse within a province. But when you change scales down to, say, the nation or the country, you'll get a different distribution because it's relative to that scale at which you delineate. So the previous slide showed you the provincial delineation, and here now you can see the national delineation. So the provincial delineation did not have so many high biodiversity sites in certain areas such as Madagascar, but when you look just at the country of Madagascar, here you can see that there's more diversity in the north than there is in the south, for example. And this allows for comparisons that may interest people that are focused on different scales of biodiversity. One concern is how this total diversity metric might compare with some very different species. These are examples with seagrass and mangroves. And you can see some of the gross patterns are similar between the total diversity, the fish and the coral, and seagrass and mangroves. But one of the big differences between these previous studies is the coarseness of the data. You can see here, for example, seagrass, there's a very large area of high diversity from southern Somalia to northern Mozambique, whereas our maps will give you that diversity at very small, much smaller scales at 6.25 square kilometers. And if you expand the maps that you saw previously, you'll actually see there is quite a bit of difference in neighboring cells in terms of this diversity. So it adds quite a bit to the spatial resolution, and we believe that it also corresponds fairly well to other metrics and other taxa in terms of biodiversity. Developed a set of criteria for priorities. One was the 3 taxas, the coral, the fish, and the combined, and the other were the scale at which we observed, at which we delineate the data. Here you're seeing, for example, the provincial priorities. So this compares all 7,039 cells in the Indian Ocean, and for each of those cells, you calculate the total number of criteria, which would be a sum of 12. So there are 12 sites, I believe, that have all 12 criteria, and those are listed in the table on the right. And this tells you where those priorities are and lists those in relative to sites with lower priorities for the provincial level. As we change scale from province to nation to ecoregion to nation, you'll see that you get a slightly different change in the priorities based on that delineation. In this case, every ecoregion has a priority because some sites will rise to the top for that by those criteria. So among the ecoregions, there are then the 11 priorities. Some ecoregions have more than one because they have more criteria. But it shows you that you can change your priorities based on reducing the scale of the delineation. Here are the national priorities. All countries, therefore, have their own representation. Many of them are countries that would be excluded from a provincial or ecoregional priority. For example, you can see Mauritius, Seychelles, South Africa now have their own priorities based on the national prioritization process. These are locations where reefs are highly clustered, and you can see many of them are located in similar areas to the provincial selection. That is southern Kenya, some sites in Tanzania and Mozambique, but also including Comoros. And this is largely where there is high densities of reefs. Interesting that Madagascar does not have many high densities of reefs. They're more sparsely distributed and therefore fall out of the clustering analysis. Our region, our province, at different scales, we can now compare it with past prioritization activities. If we pool all of those activities into the same scale that we work with, there are 91 locations that were identified, of which 6 were agreed on by all reports. Our method agreed with 3 previously identified locations, 65 aligned with 1 past report, and 28 are entirely new locations. When we then compare our locations with the database on marine protected areas, there are 208 marine protected areas in this province. We find that only 34% of our sites overlap with the existing MPA designations. Many of these differences in the past prioritizations have to do with the decisions made as to what species or what conditions, whether it's remoteness or reproduction, or whether you can see the species or not. So that some of these differences are accountable based on that. The difference between the MPAs and ours may be because many past MPAs have used other criteria other than total biodiversity, and therefore there's a considerable opportunity to increase the number of MPAs if biodiversity is considered one of the primary goals of the designations. Do not show some of the details, so there are in our papers and reports more detailed maps of countries like Kenya on the left and Madagascar on the right. These may be useful for assisting particular country designations and prioritization processes. In summary, these are the locations that fit 12 criteria, and then you can see where they're located in Tanzania, Mozambique, and a few in Comoros. These are probably the highest priority locations by a number of methods, that is clustering, species richness, etc. So these are high priority areas. Some of them are contained within the East African marine priority areas, also within the World Heritage, but at very large scales, and also some of them within the Southwest Indian Ocean Fisheries Project. But there are also a number that have not been identified previously. We decided to take this work a little bit further to look at what the future might look like for some of these locations, because although we can develop priorities based on past empirical data, one question remains is how resilient will these locations be in the future? We had previously studied coral cover distributions with a very similar method throughout the region, and what we had done there as well was use the IPCC reports and models, what are called CMIP models, to project those environmental variables into 2050, and to look at how things such as coral cover would change across that 30-year period from 2020 to 2050. And these are examples of some of the maps. They depend on the scenario, whether there's a business as usual scenario, which is the RCP 8.5 version, which assumes no carbon emission reductions, but just a continued release of carbon. And then there's the carbon emission reduction scenario here, or scenario 2.6, which assumes that there's an effort to reduce carbon emissions and keep global temperatures down. So these maps of coral cover can give you some sort of indication of where the coral cover is located in 2020 and in 2050, and then you can take the difference between them to look at the changes that occur across time. So what we did was compare this coral cover predictions that was already published with the changes in biodiversity using the same approach. That is, the variables that overlapped between the IPCC models and our models, which are 5 variables of temperature. We then modeled the predictions for numbers of taxa in 2050 in all of these 7,039 cells. This is a scatter plot showing the coral cover resilience and the coral generosilience. That's just simply the change over time, and distributed among the various countries under these 2 scenarios. And you can see that the business as usual scenario, as well as the RCP 2.5, most of the sites lie in the lower left quadrant, suggesting they're losing species and coral cover, but there's broad distribution throughout the area. We also look at local resilience, which is the top graphs, which is the comparison of each cell to itself, but then we compare cells to the average coral cover and number of species in the province, and that gives you sort of a larger perspective on the changes over time. And you can see that that changes the distribution somewhat, such that some of the there's more representation in the more improved outcomes. For example, you can see here Mayotte is shown to have, at the provincial level, fairly good outcomes of maintaining its species richness and coral cover over time. And all the other countries have some representation within each of these different quadrats, but there are definitely winners and losers with climate change. You can then map out this resilience for those 4, for the 2 scenarios, and the local and provincial resilience, and these are the kind of maps you can see which are the most resilient locations. It's quite variable in the sense, but there are a few locations, such as southwest Madagascar, which has some locations that are fairly resilient to climate change. That's true of the offshore islands, of the Mascarene and Tromelin Islands, that they are fairly resilient locally, but because they have low diversity, they often score lower in terms of the provincial resilience. Much of the African coastline is predicted to lose species and coral cover. The loss of coral cover is generally greater than the loss of species. If, for instance, in the RCP 8.5 model, the region will lose about 35% coral cover, whereas they may lose about 8% of their species. One of the overriding goals of this research is to work globally, that is, to find climate sanctuaries on a global scale, and to increase efforts by the nations, the global organizations, the NGOs that are involved in conservation of coral reefs. There has been previous efforts, such as the 50 Reefs program, to identify these areas. The 50 Reefs program has identified a number of reefs. The method they use is the excess heat method, which is the project the amount of excess heat in 2050 or 2100 and identify those areas with the lowest excess heat. This method is good for identifying refugia that will have low exposure or of what we call avoidance sanctuaries, but there are also other types of sanctuaries that are sanctuaries because corals recover quickly or they resist heat, and these might be identified by other methods. If you simply look at other types of models, such as the variability models, of which this is one of the variability models, you can see there's quite a bit of coverage of sanctuaries by this different method. In fact, if you combine these two methods and some of the efforts to identify climate refuge, you find that 30% of the reefs are actually in some sort of a refuge. So the idea here is to identify these, develop the best models, make sure that these models are effective at predicting sanctuary, and then following up with the prioritization and reducing threats to these particular high biodiversity, high sanctuary areas. I'll finish with a few recommendations to improve our attempt to find high biodiversity areas and to increase efforts to manage them well. One is that to remember is that when you change the scale of delineation, you expand the priorities in the spatial portfolio. That's shown here when you go from province down to ecoregion to nation, and you find that you have additional sites that are priorities, and many of these priorities are important, particularly for nations that may fall out of the ecoregional or provincial prioritization processes. There's a need to expand the portfolio of MPAs to include both the biodiversity and refugia areas within them. As shown here, only 34% of the 208 protected areas in the region are actually protecting high biodiversity locations, and yet we haven't even looked at the correspondence with sanctuaries. Reconsider and expand the metrics for choosing the lowest stress, most resistance, or fastest recovering corals. There's a need to look at metrics associated with particular types of refugia. There are resistant, fast recovering, and avoidance refugia. There may be specific environmental metrics that predict those particular types of refugia, and that has not been investigated. Continue to use modern statistics, artificial intelligence, and machine learning algorithms. Up until very recently, these have not been used for these types of activities, and yet they can be very useful, and there's much we can learn from these modern techniques. Augment satellite-based coverage with local knowledge of coral survival. The satellite data and information is wonderful, but in many cases we need to make sure that local knowledge can either refute or augment that particular predictions for coral survival. Include more representation of resistance and recovery refugia to balance the current avoidance refugia focus. So, for example, the 50 reefs is based on avoidance criteria, whereas there are reefs that are recovering and or resistance that won't be identified by excess heat and avoidance metrics. Find outliers, such as locations with a mixture of high coral cover, diversity, and adaptive potential to high exposure. Outliers are often very useful indicators of some unique capacity of the corals or some unique environment that allows them to survive. Finding outliers can be very useful in expanding knowledge, and that knowledge can be very useful for making management recommendations and identifying priority sites. Continue to collaborate in sharing field data and experience to compile robust, standardized, and comparable data sets. This is something that allowed this project to occur because there was a group of investigators, not a large one, but a small one, that shared their data, and we were able to get very robust models because of the high replication. And finally, to share lessons of success and especially failure broadly. There's a lot of failure that will go on during this period of learning around climate change, and yet we shouldn't shy away from sharing that information, because it's through these failures that we learn to get better at success. I'll finish there and thank USAID DOI very much for their support for this project. We believe it's been very insightful and useful for some very important activities like biodiversity identification and sanctuary identification. I was all very grateful to my coworkers, Witta, Maxwell, and Carol. And also we're very grateful to the people that shared their data with us. We will be submitting papers to journals for journal publications, and those should be coming out in the next year. Thank you very much for your attention. It's greatly appreciated.